Okay, gentlemen, so what you're going to do for this assignment is just click on A20, protein folding. You're going to watch this video, and then I want you, after you watch the video, to see if you understand how proteins fold. So we're talking about protein folding. And as I explained to you in class, proteins are very diverse in structure because they could take so many forms. And the reason they could take so many forms is there's so many different amino acids that affect the way they fold up into a three-dimensional shape. So that's what we're trying to explore today. So I gave you this web link. This is to hemoglobin. And you can click on this full 3D viewer. So I want you to be able to identify primary, secondary, tertiary, and quaternary structures in this particular protein. So how we're going to figure out the amino acids is if I click on the protein any spot. So there's 4 proteins in here signified by the different colors. So if I went to this blue protein and I wanted to know what amino acid was at spot 16, it tells me this glycine. If I wanted spot 8, it tells me it's lysine. So what I want you to do is I want you to pick out 4 amino acids, and you can either screenshot or just tell me in an explanation. So you will have to take some screenshots of this picture, but you can either just say, okay, here's 126, there's the amino acid. So in this ribbon model, they don't show you the chemicals, but the model just says, hey, these chemicals are part of a ribbon that are folded up. So you could just click on 4 of these different spots. You could take screenshots if you like. Let's go to the first protein, or the first amino acid in a protein. So let's try to figure out the spot here. So that's probably the first amino acid, and of course it's not showing, but that's how you would figure out how we get the three-dimensional structure. So let's go to the 3D view again. That's how you figure out your primary structure. So let's go back here. I said I wanted number 1. It's probably right there. Of course, it's not showing it. I might show it over here. So that's the last one, 140. Let's see if the first one. So that's 145. But you just have to pick out 4. Now, for the secondary structure, what happens in your secondary structure is you have one of 2 shapes that are taking, really 3 shapes. Either a turn, an alpha helix, or a beta pleated sheet. So those ribbons, the straight line starts to fold on one another. So let me show you an alpha helix. There's one right that I'm zooming in on. So right here, we have an alpha helix. The strain of line starts to go into this folded shape. Okay. Also, a beta pleated sheet, it's tough to see here, a beta pleated sheet. They usually zigzag across. So let's just look for beta pleated sheet. And the image you could see right here in this beta pleated sheet is just going to go back and forth. So you're just going to identify beta pleated sheets. Oh, by the way, we're looking at hemoglobin, which is a protein on the red blood cell. And for the tertiary structure, there's 4 different tertiary structures here. They're signified by the color of each amino acid. So this protein has tertiary structure. And quaternary structure is when the proteins team up together. So this has quaternary structure because there's 4 specific amino acids. So just identify the primary, secondary, tertiary structure by using this protein and hand it in. Good day.